viruses, viral infections, bacterial infections, mutations, of course, because the basis of all cancers are a genetic change and a mutation. For example, changes in oncogenes and the tumor suppressor genes, which one very good example is the breast cancer gene, which basically is an oncogene. Once it's mutated, it causes them. So that is something that was a very interesting finding from our results. We lost the slideshow, Dr. BETKAR. So so you need to take it back and bring it back, please. But let me tell you, I can continue as he's uploading the presentation. What we found as a very interesting result is when we stopped thymoquinone treatment and we let the mice survive for another 10 to 15 days, the tumors did not regain their growth capacity. So it's kind of very interesting that once you block tumor formation, so we can go to the, yeah, great, bravo, thank you. So go down to the graph so that I can show them what I mean by, yeah, this is the graph that showed that in 5FU treated 5FU, this is coming from sensitive cells, but we found also the same in resistant cells. This is the size of the tumors in the control animals that were not treated with thymoquinone. Look at the magnitude of inhibition in animals that are treated with thymoquinone. And the cause of this death mechanism was found to be induction of programmed cell death or apoptosis. Okay, what we call apoptosis. So it's kind of very interesting that we were able to identify the mechanism as programmed cell death. Not only that, but also very interesting, there was an inhibition of stem cell marker CD44. This is a CD44 slide, which shows control cells that have a lot of CD44. And, those that are treated with thymoquinone, you see a great inhibition, no greenness. So meaning that there is great inhibition in CD44, which is a stem cell marker. And this happened more so in the resistant, in the 5FU, 5-fluorouracil resistant cells, more than the 5FU sensitive cells. So that's kind of a very interesting finding. Now, next slide, I was telling you that even when we stopped thymoquinone treatment, notice here is where we stopped thymoquinone treatment, the tumors did not grow back. And look at the growth of the tumors in the control animals. So the tumors did not grow back, and that means that there's some kind of a long-term tumor inhibition in animals. That was a very interesting finding, which we published. Next slide, please. Okay, now there are many papers in the literature that show that it is better to combine thymoquinone with clinically used drugs. So there is, for example, data on combining it with cisplatin. There's data on combining it with doxorubicin. There's data on combining it with 5-fluorouracil. So it's better to, you get a more enhanced effect if you combine thymoquinone with clinically used standard drugs in patients, okay? And there's numerous literature. You can just put thymoquinone and combination therapy, and you can find hundreds and hundreds of papers that have shown enhanced efficacy of thymoquinone when you combine it with clinical drugs. Now, what's the advantage of this combination? The main advantage of this, that's great. Now, the slideshow is excellent. So the main advantage of this combination is that clinical drugs are toxic to humans. So when we combine thymoquinone with clinical drugs, we are able to reduce the dose of the clinical drug to non-toxic levels by this combination. That's why this combination therapy has gained a lot of attention, because you're able to reduce the toxicity of the clinically used drug by combining it with a natural drug like thymoquinone. Next slide, please. Now, thymoquinone, this is a review that we have published recently, about, a couple of years ago, which shows that, thymoquinone has been combined with so many drugs like cyclophosphamide, like, cisplatin, like I told you, 5-fluorouracil, paclitaxel. So if you are interested in combination therapy research, then read that review paper. It gives you a lot of insights on how we were able to combine thymoquinone and get greater efficacy and less toxicity. Next slide, please. Okay, so the advantages, like I said, is the augmentation of its anti-neoplastic effects, of the anti-neoplastic effect of the chemotherapy drug. Okay, improvement of outcomes of cancer resistance. Also, there is less cancer resistance when you combine thymoquinone with clinical drugs or conventional drugs. Basically, we are able to lower the effective dose of standard chemotherapy and get the same efficacy, and we reduce the toxicity of the standard chemotherapy drugs. So that is, the main reason why this combination therapy has gained a lot of attention, as well as basically maintaining the effectiveness of the standard chemotherapy. So what's interesting is when you combine thymoquinone with the chemotherapy drug, you get the same efficacy as the standard chemotherapy drug. Okay, so if we can go to the other slide, next slide, please. So not only have we combined thymoquinone with the chemotherapy drugs, we have also combined it with natural compounds. So for example, lots of natural compounds like vitamin D, like melatonin, like resveratrol. Okay, so there are many compounds that have been, genistein, for example, that have been combined with thymoquinone, and that is also reviewed in our paper. Okay, the review paper shows how thymoquinone was combined with clinical drugs as well as with natural drugs to inhibit cancers like colon cancer, leukemia, breast cancer, and many cancers. So this is a paper that was published in 2021 and in the World Journal of Clinical Oncology, if you're interested in, just looking at it or reading it. Okay, next slide, please. Now, this is where we've been working with this molecule for 25 years, not us only. Now, a lot of Westerners are interested in this compound and its effectiveness. There is a lot of data from US labs, from European labs, Germany, Sweden, you name it, Chinese, Indian. So there are a lot of, there's a lot of research from the international community. There's a lot of interest in this molecule. It is now 25 years since this molecule has been researched and published on. And the big question then becomes, why haven't we translated it to the clinic yet? Sometimes molecules don't take that long. It takes, like I said, around 10 years, sometimes 15, sometimes max 20. But this has exceeded the 20-year limit. And the reason for that is that this molecule is very hydrophobic. Now, hydrophobic molecules are hard to target to tumor cells. So we need to be able to find a way to either encapsulate it and make it more hydrophilic and reachable to the tumor site. Not only is it hydrophobic, but I have done so many experiments in animal studies to be able to capture it in vivo. And I'm unable to do that even if I use it at very high doses. Somehow, once you inject this compound into the bloodstream, it disappears. It disappears and you cannot find it. Who takes it and who takes it in the cell? Who carries it? And who takes it to the cell is not yet known. So is it complexing with another protein that acts as a carrier, takes it into the cell? Does it have a receptor on the surface of the cell membrane? What is happening to this molecule? There are zero pharmacokinetic and pharmacodynamic studies on this molecule. And unless you are able to do PK, PD studies, pharmacokinetic and pharmacodynamic studies on a molecule, it will not reach the clinic. Okay, so I have there's a lot of promise in this molecule, but there are lots of challenges because of its hydrophobic nature, and we don't know how it gets into the cell and how it exerts its effect on the cell. And this is recent research. This is hopefully the work of my next master and PhD student, is to find out who carries it. Okay, so we're looking into that, and hopefully we will have an answer soon. Okay, next slide. Okay, now another thing that you can do is, since it is very unstable and since we cannot find it in the bloodstream, is to encapsulate it, is to nanoparticle encapsulate it and improve its drug delivery. And there are so many drug delivery systems that have come into the market, and sometimes some of them they are in phase 1 and phase 2 clinical trials with certain chemotherapeutic drugs. So the paclitaxel, for example, has a delivery formulation. There are many drugs that have delivery formulations, but not as much as we would like to see. We would like to see more of these formulations. Nanoparticle encapsulation is not an old science. It's a more recent science. So the advantage of encapsulating thymoquinone is we would improve its oral absorption, its stability. We would probably protect it from gastric breakdown, proteolytic enzyme breakdown. We would probably also improve its bypass through the liver system, through the liver enzymes, the breakage through the liver enzymes. We would probably increase its therapeutic potential, hopefully have higher concentrations in the blood plasma so that we can detect it. So there are many advantages for encapsulating, and we have published some work on nanoparticle encapsulation of thymoquinone, but whatever we have published is not good enough for animal studies. So that's why we are looking into new formulations recently that could be appropriate for animal work, because the formulations that we have published on are somehow toxic to animals. They kill the animals. So we need better, safer formulations. Next slide. And this is what we're also working on currently. Now, one of the formulations that we worked on, which is not unfortunately good for in vivo work, but it is excellent for in vitro, and we found lots of interesting data on this, is the cubosomal formulation. So it showed a lot of promise in terms of being biocompatible, biodegradable, lipophilic. It could hold the thymoquinone. It had a high drug loading capacity. It had sustained releasing behavior. And we hope that it will have in vivo stability, but unfortunately, the in vivo cubosomes were, not very safe to animals. So that's why we stopped research on the cubosomes, and we are looking into other formulations. Next slide. They're very simple to prepare. So this is just what we did with the cubosomal formulation against breast cancer cells, and very resistant breast cancer cells. MDA and B231s are very resistant breast cancer cells, and we were able to show that it is better than free TQ. It targets those breast cancer cells and inhibits their viability more so than free TQ. Okay, next slide. So another good formulation or nanoparticle method of encapsulation is to use phytosomes. Now, phytosomes are interesting drug delivery systems, which we can, we hopefully will be able to explore with our collaborators. I'm not a nanoparticle expert. I collaborate with researchers from different institutions to basically be able to do this research. But the interesting thing about phytosomes is you can encase the thymoquinone in a lipid vesicle that is a double membrane which can fuse with the cancer cells. So that's the interesting thing about using phytosomes, is to basically encase thymoquinone in a double lipid membrane, which can fuse easily with the cancer cells, with the membrane of the cancer cells, and deliver the drug to the cells in a steady, hopefully a slow release. Next slide. These are some commercially available phytosomes for certain plant products. So they have been, for example, used against flavonoids that come from this plant, this interesting plant. They have been used against ginkgo biloba, okay, which is a very interesting plant that has been shown to have a lot of anti-inflammatory effects. Many of us use it as a supplement to inhibit inflammation in the body, ginkgo biloba, and also it is known to be very good supplement for increasing memory. Okay, so it can also enhance memory in humans. So it's a very interesting plant that has been encased in or formulated in phytosomes. Another very interesting compound that has a lot of applications. I haven't researched this compound, but there's numerous publications about this compound in the literature, which is curcumin. Curcumin has also been encased and has been formulated in phytosomes, and has shown after formulation to have close to 30 times greater activity. So you can see that you can increase the activity of the drug after you protect it in some kind of a formulation that can, deliver it slowly and protect it from being degraded by enzymes. We can go to the next slide. So the next, basically, is the next effect that I want to talk to you about is the anti-cancer potential of phytosomes in preclinical studies. Like I told you, certain phytosomes have been used in preclinical studies, and some of them have been also used in clinical studies. So this is a plant which basically has been, look at the compound. The compound is very large and very kind of, it's a polyphenolic compound. So it's a very large compound, which once encased in phytosomes, it can target to a better extent ovarian cancer cells. And it's more potent and more cytotoxic when it is formulated. So there is hope. The challenges can be overcome by basically encapsulating these compounds in different kinds of formulations. Next slide. I'm getting close to finishing up. These are other examples that, olive oil, black seed has also been put in phytosomes. Olive leaf compounds like eleutherin and rutin have been also placed in drug delivery systems for better activity. Next slide. Immunotherapy is a very interesting way to target cancer cells. What you can do with this, what is cancer immunotherapy, is to basically prepare cancer vaccines that can boost the immune system. And as a result of this boosting, the body's own defense mechanism can target the tumor. See, the problem with tumors is they mask your immune system. Why are they able to grow? Tumors are able to grow and proliferate and increase in size because they mask because they're able to mask the immune system. So if you can unmask and boost your immune system to target the cancer cells, that is what cancer immunotherapy is all about. Development of cancer vaccines. Not many have been developed and not many are used in the clinic. But if we are able to, the main problem is the delivery. The main problem of cancer vaccines is where do we inject them? So the best methods of injection has been shown to be subcutaneous, but they're not very stable. Unfortunately, they're not very stable. But this is a talk that is still, or this is a conversation that is still happening in order to improve cancer, treatment. Immunotherapy is a way to improve cancer treatment. There's a lot of research happening lately. Okay, there are some peptide vaccines that have been developed. Okay, these are peptide, vaccines that you can inject into the patient, and you can basically boost the immune system and target these peptide vaccines sometimes to the cancer cells. Okay, so you can target the T cell, which are the cytotoxic T cells, will be able to kill okay, the cancer cell. Because what this peptide does, what this peptide vaccine does, it presents itself, okay, on antigen presenting cells. And once it presents itself on antigen presenting cells, the T cell recognizes it as basically being foreign and attacks the cancer cell. That's the main idea, is to take this vaccine, put it on the surface of antigen presenting cells and boost the immune system and have the T cells basically be activated in order to kill the tumor cell. Okay, next slide. Okay, these are basically adjuvants. We have also been able to use some adjuvants like curcumin as an adjuvant for cancer vaccines. So not only do we would we want to boost the immune system by cancer immunotherapy, but we would also want to combine it with adjuvants. Now, what are adjuvants? Adjuvants are chemicals that come from natural sources that could be used in addition, in combination with cancer vaccines. Curcumin being one of them. Another very interesting adjuvant is QS21, which comes from this interesting plant, and it's a saponin molecule, which can be used in combination with cancer vaccines. Next slide, please. Next slide. Okay, so these adjuvants, what are the advantages of adjuvants? Basically, they can be, for example, they can facilitate the immunization for apoptosis induction. They can have an effect on antigen presentation. They can enhance cell death and cell modulated mechanisms and immune mechanisms. So these adjuvants can help the cancer vaccines to basically boost their effects, further. Next slide. So in conclusion, basically targeted therapies, I believe that targeted therapies, if not personalized medicine, if not precision medicine, because of the cost for the market, for the companies to make precision medicine medicines, or to make individualized medicines, if this is a cost to the companies, maybe targeted treatments, I think they will be predominating the 21st century. And plant compounds have been a very important source. And despite the new approaches to drug discovery, like combinatorial medicine and computer-based medicine, they're unlikely to replace the important role of plant species. And to reduce the challenges of medicinal plant applications, the best way to do it is to hopefully encapsulate them or deliver them in a better delivery system to make them more stable and to release them in a sustained way over a longer period of time. And I still believe that Hippocrates, basically Hippocrates 25 centuries ago said, "Let food be thy medicine and medicine be thy food." So that's a very interesting saying that we can probably benefit from what we eat. And I would like to thank you for your attention. I'm done. If you want to ask questions, I will be happy to answer.
Thank you, Dr. Hala, madam. It was very nice project. It was very nice presentation, useful presentation, and your research work, your hard work for curing the cancer is very, very appreciable. And you basically have used the drug delivery system, the plants which is having potential of curing the cancers. So these things are very, very useful one for the human health. Thank you for you. And I welcome our director of the Veer Maheshwar Charitable Trust, Sri Veerbhadracharya Ganacharya. Salute to you, sir. And today is the 100th day, 100. It's the golden jubilee. Hello. Yeah. For this continuous session, and we are very fortunate that your topic on this today's era, it's very useful. Now I welcome all the participants can unmute and discuss with our resource person, madam. First of all, I would like to welcome Dr. Swanand Shukla, sir. he is doctor, and so he can discuss very clearly with you. Thank you, sir. Welcome, please.
Good evening, Dr. Betkar, sir, Principal, sir. Guruji, namaste. And hello, madam. I think it is a good afternoon to your place. So, I think, what Hippocrates had said that the life is short and the art is long. So, I think the art means our science is, so vast, we cannot gather it in one lifetime. And whatever we are having is the best of the past. And that is what we are carrying forward. So every tradition, every culture, every geographical region has its own medicines. And the traditional medicines, what we call it as, very recently, means just 3 days back, the WHO, there was a summit, Ayush summit, Ayush in the sense Ayurveda and all traditional medicines were included. And today itself, India's Prime Minister, Honorable Prime Minister, concluded, with his speech in a valedictory function about this all aspects. So it is really wonderful thing that we are coming with something new, because we know in cancer, the chemotherapy and radiotherapy, these two therapies are there, and we know they are abundant. There are side effects. And no doubt, we have to find out some alternative medicines. So wonderfully, you have placed whatever the studies you have done. It is very great thing. And I think you have done some animal study also. But, this traditional medicines requires a kind of support of, study like randomized control trial, evidence-based, studies is very essential, because that is how it will become universal. Otherwise, people
Yes.
may consider, people may not reach to these medicines. So that we have to conduct randomized control trial instead of some animal studies or vitro, studies we are doing. But I think we have to go with, we have to do a trials on the human beings, and whether, we can compare it with the conventional medicines. And that is how we can draw inference from it. So that is what is required. I am a basically homeopathic physician, and we are also treating cancer patients. And most of the times, we cannot insist patient to leave the other medicines. So we are going with what is called as a, um, excuse me.
Okay, if I can comment before he comes back. Okay,
Yes.
you're back. Yeah,
Yeah,
go ahead.
I'm back. So I think, this kind of studies are required. I'm a homeopath. We are using medications, but we are going with add-on therapies, and we are also publishing our some papers. But most of them are add-on therapies, because people strongly believes in other, medications, and it is their right to select what kind of therapy they have to go with. And no doubt, we what you were talking about, or Ayurvedic system of medicines are having medicines. Unani system is also having medicine. Chinese, they are also having their own medications. But very important thing is, we should have evidence based, and that is how it will be accepted. And it is one of the very important thing. It is one of the very important thing what we are discussing. Are you getting my voice?
Yes,
Hello.
we can hear you. Yes, we can hear you.
Yeah. So what do you think, we should go with such a kind of a broad scale, trials has to be done. Unless and until we are not going on a broad scale trial, we cannot prove the efficacy of our medications. So
Okay,
whatever
let
you
me
have done is a great job, and you have placed your energy in that. But it has to reach to the persons who really required it, because the patient who suffers with breast cancer, oral cancer, or colorectal cancers, right? They are the vulnerable people, and they should get some kind of therapies. And radiotherapy and chemotherapy should not be a last resort. Many of the times it seems to be that is a terminal treatment what we are doing. Another very important one that you please comment on this. And another one question is there. What about the toxicity? Many of the Ayurvedic preparations are there. we should bother about toxicity. Many of the time the people are having idea that the Ayurvedic medicines means the herbs, and herbs doesn't have any kind of toxic effect, but it is not so. So one has to think rationally, logically, and scientifically what is the kind of toxicity and therapeutic dose, what is the line in between these two. So these are the two important aspects, when we are talking about such a kind of medications. So please comment on this, ma'am. Otherwise, your explanation was wonderful. Thank you.
Thank you. Thank you very much. Thank you for these two very interesting comments, actually. In terms of randomized clinical trials. Now, let me also tell you a little bit about the disadvantages of chemotherapy. Chemotherapy not only is toxic to the body and, usually knocks the person down, and we know about, hair loss and, diarrhea and vomiting and so on. But the one, and I've seen this in many talks abroad when I go to conferences, chemotherapy can change the nature of your cancer to make it more aggressive and to kill you faster. And this is evidence that all physicians know, and yet they insist on using chemotherapy because it gives you fast results. Chemotherapy causes resistance, and not just resistance. I mean, I can give the example of my dad. He had prostate cancer, and it was stage 2-3. And he was given first hormonal therapy, everything disappeared. Then he was given chemotherapy, and he died from a very rare abdominal cancer because of chemotherapy. So, and in a very short period of time, he lost his life in a very short period of time. So although it gives you results and you can see results, and that's what physicians want to give the patient. They want to give the patient a hope that, they are getting better. But they know, physicians do know that chemotherapy in the long run, or maybe in the short term, depends on the kind of patient, it can cause greater resistance, and the patient can die from a different disease than his original disease. Okay, that's one main reason, why chemotherapy should be looked at with a grain of salt. Now, with respect to randomized clinical trials, I think the main challenge, unless they are done at the university level, in maybe, clinical units, companies are not interested in conducting randomized clinical trials of natural drugs, unfortunately. Companies don't want to do that. Okay, so I've talked to many companies about thymoquinone and their willingness to move it further. Despite its very promising results, they're not interested in investing in natural compounds because the market is limited. The market for natural compounds is limited. People rely more on chemotherapy and radiation
and
therapy.
The pharmaceutical companies are making money on it. The basic problem is this.
Exactly. That's it's an ironic thing. It's very ironic that physicians prescribe it because the pharma company is supporting it, and they know its disadvantages, and yet they prescribe it to patients. I think when they make their oath that we will have to protect humanity, they know that the disadvantages of chemotherapy, but they want the patient to get these fast results and to be happy with their cure faster, but they know that there's resistance. Now, unless we convince pharma companies to conduct randomized clinical trials, and we do it in collaboration with institutions, with universities, it has to be in clinical units, in clinical care units in universities. I don't think that we can advance this type of research, unfortunately. Okay, we have to convince pharma to basically support it, because we cannot, I'm a bench researcher, I'm a basic scientist, I'm not a physician, I'm not a clinician. I cannot, move a drug unless the pharma supports me and, is able to do all these pathological studies, PK/PD studies, toxicity studies in animals, then move it to phase 1, phase 2, phase 3.
Yes,
I cannot do it myself. Yeah, that's the disadvantage. And in terms of toxicity, I agree with you totally about the toxicity of natural compounds. We have to be able to also advise people, because people have contacted me. Can I give thymoquinone to my cancer patient? They contacting me by email. I tell them, don't do that, because we don't know what is its harmful effects on humans. We have not tested it on humans yet. We know that in animals at doses higher than 25 milligram per kilogram, it causes epileptic-like shocks. It affects the neuronal system, okay? But at lower doses, it has treated Alzheimer's. It has improved Alzheimer's in patients. So it affects the neurological system, but we have to find the relatively non-toxic dose in humans before we move these compounds to patients. And they have to be advised. Patients have to be advised that don't go out and take these compounds just because they're natural. Natural does not mean non-toxic. Many natural compounds are also toxic. Okay?
Thank you very much, ma'am. Wonderful explanation. And I think you should approach WHO, because they are promoting nowadays traditional medicines.
Okay.
So I think through their channel, we can work out with this kind of medications.
Okay, can you send me the link of their new approach, of their new
definitely.
announcement?
I'll be sending it to Dr. BETKAR. Yeah.
Yes. Yeah, I'm in touch with him. Yeah, if you can send me the link for their new approach and strategy, would be great. Thank you so much.
Definitely, I'll do.
Thank you, Dr. Sir. I will give details of her, and you may communicate further. So scientific discussion it was. And I welcome other participants. They can unmute and they can discuss with our today's speaker.
Hello.
Yes, sir.
Hello. Good evening, Dr. Betkar, sir. Good evening, Hala, ma'am. I think this is one of the best presentation about cancers and its related issues. And I'm very happy. My question is, can yoga help to reduce cancer? Thank you.
Your question is what? Can we help reduce cancer?
Yoga.
Yoga. Oh, yoga. To be honest, I'm a medicinal plant person. Yoga definitely can improve the mental status. See, a lot of cancers happen because, and with this there's really recent evidence that shows that stress causes cancer. Okay, the stress factors of life, because stress factors of life of certain patients, they have done some studies that show that stress factors are one of the reasons why cancer develops more in some patients than others. Two patients can have the same mutation. One develops cancer, one does not. Why? Because of their different lifestyle. So I think what yoga can help in, it can help in, easing the mental status, the mental stress of individuals. Probably would be acting more as a preventive mechanism than a therapeutic mechanism. I think so. Okay, I'm not a yoga expert, but I believe that definitely yoga, if it's done properly and if it's done close to heart and regularly and as it should be done over a long period of time, definitely I think it would be a very good prevention mechanism.
Thank you so much, ma'am. I'm very happy to see Dr. Veer sir. Sir, nice to meet you, sir. Thank you, sir.
Thank you.
Anyone want to discuss? Because this topic is more familiar to us. We are not from medical field, but what she explained, it's very understandable, because the plants she have used, the reactions and the characterization she have did for the curing the cancer. So it's very nice. Thank you, ma'am. And if anyone want to discuss, they can discuss, please. I think everyone here completely understood our topic, and I'm very happy that your presentation was best presentation, useful presentation, because our research is approaching towards the well health of the humans, and you have did and doing job the curing of the cancer with various plants, and thank you for that. I wish all the best outcome from your research in future. I best wishes to your all students, those who are doing study under your supervision. So it's very nice thing communicating with you and attaching this topic or connecting this topic to others. Thank you, ma'am. Thank you, please.
Thank you very much. Thank you. Appreciate it. Really, it's a pleasure. Thank you for the invitation. Okay, bye for everyone. Good evening and bye-bye.
Bye-bye.
Good evening, ma'am. Good evening. धन्यवाद गुरुदेव जी, आपका यहाँ पधारने से हम आपका स्वागत करते हैं और बहुत ही धन्यवाद प्रकट करते हैं कि आपने हमारे लिए इतना अच्छा टाइम निकाला है। आज का शुभ दिन है, हमारे यहाँ तो त्योहार चल रहा है, फिर भी सभी हम इकट्ठे हुए हैं, एक अच्छा नॉलेज मिला है हमें, तो इससे बड़ी खुशी क्या हो सकती है कि आप यहाँ पधारे हैं। आज का 100th डे ऑफ आवर प्रेजेंटेशन इज सेलिब्रेटेड इन ओनली प्रेजेंस ऑफ यू। आपकी पवित्र सानिध्य में हम आज का 100वां भाग 100वां भाग हम यहाँ सादर किए हैं। इससे बड़ी हमें खुशी क्या है। तो आपको सभी पहचानने लगे हैं, तो ये मुझे बहुत गर्व की बात है कि श्याम सुरेश सर आल्सो communicated with you, and so thank you sir, thank you sir, thank you doctor sir for nice discussion.
Thank you, sir जी.
आजची कॉन्फरन्स खूप सुंदर झाली सर, धन्यवाद सर.
सर्वांना विनंती आहे की आपण आपली फीडबॅक लिंक भरावी, जेणेकरून आपल्याला सर्टिफिकेट मिळेल. आज फीडबॅक लिंक ही तिथेच चालू झाली, म्हणजे याच्यामध्ये काय आपल्या ग्रुपमध्येच ऍक्टिव्ह व्हायला लागली. तसं काही अडचण नाही, ग्रुपमध्ये जायची गरज नाही, इथंच आपण फीडबॅक लिंक भरू शकता. ग्रुपवर देण्यात काही हे नाही, त्याच्यामधलं देण्यात काही अर्थ नाही असं वाटतंय, कारण ग्रुपवर न सहभागी झालेले बरेच भरतायत. धन्यवाद गुरुदेव जी, आपका बहुत महत्वपूर्ण टाइम रहा हमारे साथ. हमें बहुत अच्छा लगा कि आप यहाँ आज उपस्थित रहे, हमें आशीर्वाद मिला और हमें आनंद है, हमारा सौभाग्य है. थैंक यू, थैंक यू. बोला गुरुदेव, अनम्यूट व्हायला पाहिजे तुम्ही.
हा सर, अभिनंदन सर.
थँक्यू. आपले आशीर्वाद महत्वाचे आहेत. रोज जे करतोय आपण ते
नाही, अतिशय अप्रतिम गुड प्रेझेंटेशन झालं आजचं.